All right, everyone, we're going to go ahead and get started. Thank you so much for being here today. If you're on site, welcome. If you're online, also welcome. Today, we are here for our research seminar. My name is Cinnamon Moffett, and I'm the research program manager here at Hatfield, located in Newport, Oregon. And again, thank you all for being here. A couple of logistics before we get started. If you are online, we do have your mics, screen shares, and cameras turned off, but we hope that you ask us questions at any time by putting it into the chat or raising your hand, and we will be able to unmute you. We'll get to your questions or your hand raises at the end of today's presentation. For those of you in the room, we have Michael Banks here. He is the one that's going to be helping us with the questions that are put into chat. We also have Shannon Reed here, who's going to be the one helping us moderate any of the folks that are online. So for folks that are online, if you're having any technical issues, you can also put in questions into the chat for Shannon, and she will let you know how she can help you. For those folks in the room, most of you are familiar faces, but if anybody is not that I'm not recognizing, remember that face coverings are required while you're in this space. So if you need to get a drink, please step out into the hallway where there's designated eating. The other thing to know is that if you have questions, we'll get to them at the end, but you will need to have a mic. So please raise your hand and I will bring you the mic. Any questions in the room or online before we get started? Okay, a couple of other announcements I just wanted to make. Next week, we have another hybrid seminar. So that means that you can either be here on site or online. And we have Megan Wingrove, who's assistant professor for coastal engineering out of the College of Engineering at Oregon State University, and a new member of Hatfield Marine Science Center, will be coming to speak to us about beach dune subsurface hydrodynamics and the formation of dune scarps. So I'm hoping that folks come and welcome her into the new position here at Hatfield. If you need any other information about any of our upcoming events, you can log on to the Hatfield's homepage, scroll to the bottom, and you'll find a calendar of events there. I also wanted to let folks know this is being recorded. So if you're interested in watching it again, because you're so excited about our presenter, or if you want to share it with others, it'll be posted on our past seminar page in a couple of days. For folks online, that information was already put into the chat box by Shannon. Okay, and I also wanted to let the folks know that I'm starting to book seminars into the winter, and I have a few open dates. So if anybody is interested in giving a seminar or would like to volunteer somebody else, please contact me and we'll get you on the seminar schedule. For today's talk, we actually have somebody here who is working with Lee Torres. And so Lee Torres, will you come on up and introduce today's speaker?
Cool. Awesome. Great to be here in person. Woohoo! Okay. All right. Yes, it is my pleasure to introduce Casey Bierlich, who is my postdoc here in the GEMM lab in the Marine Mammal Institute. So Casey is a California boy. He did his undergrad at Sonoma State, where he studied the stress physiology of northern elephant seals. And then he took a career path kind of similar to my own. He went to Duke and got a master's degree in coastal environmental management. I did the same, and then he stayed on and did his PhD there at the Duke Marine Lab, as I did, so a lot of similarities. Let's see, for his master's degree, he studied the skin microbiome of Antarctic humpback whales. Pretty cool, so he has a diversity of expertise besides drone stuff. But he really got into drones while at Duke, where he worked in the Marine Robotics and Remote Sensing Lab with Dave Johnston. And he spent a lot of time in Antarctica, studying the whales there and their body condition and behavior, and also has done a bunch of work in California with colleagues there, combining drones and tagging data. So he describes himself as he uses drone-based photogrammetry to study ecomorphology and body condition of baleen whales, and has developed several open source photogrammetry tools to help researchers obtain accurate measurements. So I think he's going to tell you a lot about that today, and his real quantitative approach to using drone photogrammetry, which is awesome, which is part of why I hired him as my postdoc. The other part is really, I was impressed when we met and had many discussions last year before I hired him, about his, he really has a great understanding of putting all this quantitative body morphology information into an ecology perspective, and better to trying to use that information to help us better understand their life history patterns and their foraging behaviors and how they're adapting to change in their environments. So he's got a well-rounded perspective, which I really appreciate. The other thing I'll tell you about Casey is he started in a flash here. He started June 1st, and I think within 3 days later, he was on a boat flying drones for us over gray whales here, which is the majority of his work, while here is helping us with our massive drone data set on gray whales. So I'm sure he's going to tell you some about that now. So welcome, Casey, to the Marine Lab, and take it away.
All right, can you guys hear me? Sweet. Thank you so much, Lee. That was a great introduction. I know every speaker probably starts off talking about how great it is to be in person, but it's awesome. This is great to be here in person. This is amazing. This is amazing space. It's great to be here. I feel like we should do like the wave or something across, you know, there's a good distribution of people across the aisles. But yeah, it's great to be here. Like Lee said, I started here in June. I've had a blast working with her in the GEMM lab. I know there's a lot of folks I still haven't met yet, so shoot me an email. I'd love to meet. The cafe is open. Let's grab coffee. Let's chat. Today, I'm going to talk a lot about the work I did at Duke during my PhD in the Marine Robotics and Remote Sensing Lab. It sets a lot of the foundational work that we're doing here in the GEMM lab now, and so I'll kind of touch on that work and then touch on the projects that we're working in the GEMM lab to kind of take those to the next step, which I'm really excited about. So morphology is one of the most fundamental aspects for understanding a species ecology. And past studies have shown how fish body shape and fish fin shape can predict and influence habitat use, foraging behavior, physiology, and overall performance. And so these different traits can impact the success of an animal and its environment. And it can show the current health of the individual and its likelihood of survival and its potential for reproduction. And if you can scale this up to analyze the population level health, which then in turn can be used for conservation. And a good example of this is the European eel, where skull size was directly linked to different ecomorphs, which have different prey types, which then the different prey types have different pollutants or toxin levels, which then some ecomorphs have lower reproductive success. And so this framework is really useful for not only understanding a basic ecology of a species, but you can also use it to have direct implications for conservation. And so how do you adopt something like this for whales? They're often found in remote environments. They spend little time at the surface, and their large size makes it very difficult to get measurements from them. You can't just dive in the water and ask them to hold still and get a measurement. Are we all good? Sure. Oh, what's that?
Is that better? Yeah.
Oh, all right. Let me take the jacket off. The jacket is coming off. All right, is that better? I did not know what I was signing up for. Wow. Okay. So so how do we adopt this framework for whales? They're large and difficult to study. And this is important because as marine mammals serve as top predators, they're ecosystem sentinels. So they not only reflect the health of their populations, but the health of the environment. And a lot of studies recently have expanded upon this idea to look at the larger role whales play, particularly in the carbon cycle, and thinking of whales as bodies of actual carbon storage and how they can be used to help mitigate climate change. And I was on a paper recently with Matt Savoca that we just published in Nature a couple of weeks ago, where we looked at prey consumption rates of baleen whales, but we also addressed the role that whales play in nutrient cycling, and that the oceans probably looked a lot different before we removed over 2 million whales from commercial whaling. And so restoring whale populations to numbers that were similar to pre-whaling numbers could help augment productivity and restore ecosystem function. And so despite the hero-like role whales play in our environments or in the ocean, there's threats to their recovery. Study recently came out showing that species, marine animals show greater response to climate change compared to terrestrial animals. And we definitely are starting to see a lot of those effects. And so the recent advancement of drones or unoccupied aircraft systems or UAS has provided immense opportunity to be able to actually study these animals in a new perspective and actually obtain measurements of them. And so we applied drones to this framework. Now all of a sudden we can get better morphology measurements and start studying these animals. And a lot of studies have been using this new technology, looking at different morphology to be able to compare different subspecies and identify different subspecies, compare body condition of animals across the season and compare it to individuals, and even looking at comparing tag data with drones to look at the biomechanics of how an animal actually moves and performs its behaviors in its environment. And so it's a really exciting time to be in this field, as I feel like we're able to ask a whole bunch of new questions that we haven't been able to ask before and learn a lot about these animals. However, there's a lack of standardization and interoperability amongst these projects. There's various drone platforms and sensors being used, drones equipped with different cameras, focal length lenses, altimeters, which measure your altitude. There's various photogrammetry software. There's several different approaches for measuring body condition, and there's no unified way to predict photogrammetric uncertainty across this methodological spectrum. And this last point is particularly important, as the National Institute of Standards and Technology, or NIST, says a measurement is complete only when accompanied by a quantitative statement of its uncertainty. And so this creates confusion over pros and cons of using one drone platform or method over another, difficulty in determining if results are reliable. An example, are you actually measuring a true change in body condition? This makes it difficult to compare results across studies, hinders collaboration and interoperability, and there's no clear route for researchers to implement best practices. And so my PhD focused a lot on answering some of these questions, or trying to answer some of these questions at least. And so my objectives for the rest of the talk, I'm going to give a quick overview on drone-based photogrammetry and how to incorporate measurement uncertainty associated with different drones using a Bayesian statistical model. I'm going to compare different methods for measuring body condition of baleen whales from drone imagery while incorporating uncertainty with each method. And then I'm going to provide a case study on how to use these techniques to measure body condition change in humpbacks along the Western Antarctic Peninsula. And then I'll talk about some projects that we're working on in the GEMM lab, kind of using these techniques and applying this framework. So we'll dive right into it. We recently published this paper in the beginning of summer. This is from my PhD, and some of the work lays the foundational work for the uncertainty model. So when we collect imagery of a whale, we often do hand and launch recovery from small boats. This is in Antarctica. You can't see the drone, but you can kind of see a humpback whale waving out in the distance there. And so I'm flying the drone over the whale right now. You can kind of see in the screen, there's two whales in there that I'm able to see and collect images of it. When I feel like I got enough good images of the whale, we'll safely land it back in our advisor's hands. And sometimes we get fun images like this, where the whale's doing some sort of water ballet. And when we collect an image, there's two main sources of error that creep in. One's from environment, the other's from animal behavior. Environment can include stuff like refraction or watercolor, being unable to detect the edges of the whale, or glare from the sun. And so we can control these to a certain degree by filtering for high quality images. So we aim for images like the ones on the right, where the animal is straight, either at the surface or very close to the surface, where the edges of the whale are very easy to see. And so once we have our image of a whale from a drone, we know the sensor width and the focal length of the camera. We know the image width in pixels. We get the altitude of the drone when it took the image, and we use those to calculate the ground sampling distance, or the GSD. And the GSD is the distance each pixel represents on the ground. And it essentially sets the scale of your photo. And once you know the scale, then you can make a measurement in pixels and then multiply that by your scale to get a true length. And to help with the actual measuring process, I co-developed Morphometrics, which is an open source photogrammetry software during my PhD to help with measuring and make it more standardized. It's really easy to use. You can measure angles and areas and lengths on the whale and divide the whale into widths to be able to measure body condition, which I'll talk about in a minute. And so coming back to this, when we look at the GSD equation, once you choose your camera on your drone, your sensor width, image width, and focal length are all fixed. So your altitude is going to have your biggest impact on GSD. And so if we plot altitude on the x-axis and ground sampling distance on the y in centimeters per pixel, and each color represents different drones that are commonly used in studies. So different drones with different cameras and focal length lenses and sensors. You can see that as altitude increases, the ground sampling distance increases at different rates for different cameras, which means that higher GSD equals a more pixelated image. It's a lower resolution. So it's harder to detect edges and objects in an image. So there's this inherent difference just by the camera that you choose based on altitude. And so altitude is a major source of error for getting a measurement. And altitude is measured either a barometer. All drones have a barometer. It's considered to have low accuracy. And some drones are equipped with a laser altimeter, which is considered to have high accuracy. The actual measurement itself is also a place where error can creep in too. So I wanted to develop a Bayesian statistical model that jointly takes the errors from altitude and the length measurement, as well as that can jointly take errors specific to a barometer and a laser and combine them as well. And so to do this, I took an experimental approach where I measured a known size object floating at the surface. I flew 7 different drones with different cameras and focal length lenses. I flew them between 10 and 120 meter altitudes. And I used the known sized object to back calculate to get the error of each drone at each altitude. And so this can be then used, or actually, yeah, jumping ahead. And so this is what that looks like. We got percent error on the y-axis and each drone on the x. We have measurements of the known size object using a barometer in red and using a laser in this green-blue color. And the thing that stands out is using a laser altimeter dramatically reduces your error across the board, even for the last 2 drones, which are the DJI Phantom 4 Pros, which are off-the-shelf commercial drones. And so this is really useful. We can use this as training data now into the Bayesian statistical model. And so the Bayesian approach views the data and the parameters that generate the data as random. And so you can describe parameters by a statistical distribution. And so you have your likelihood function, which is a model of the observed data. And then you have prior knowledge about the underlying parameter that you can incorporate by using a prior probability distribution. And then you can use Bayes' theorem, which combines the likelihood and the prior probability distribution to then generate a posterior distribution, which serves as updated knowledge about the underlying parameter. And so this is actually mimics how we think and how we learn. If I told you guys I saw a 90-foot blue whale, you would use your prior knowledge on what you know about the lengths of blue whales as being, okay, the largest animal that's ever lived. And you would conclude, oh, wow, that's awesome. How majestic. I'm so happy that you had to experience that. But if I told you I saw a 200-foot blue whale, your prior knowledge, you'd use your prior knowledge and you'd generate a posterior distribution that would conclude that that statement is probably false. So using that framework, we can take the known size object that we flew different drones at different altitudes, and we can generate a posterior distribution for error. And then we can use that posterior distribution for error as our new prior information for when we're flying over an unknown size object like a whale. And we can generate a posterior predictive distribution for length. And so I tested this then on 52 humpback whales and took their length measurements. And so rather than a point estimate for length, we now get a distribution for each individual humpback whale. And we can describe their length by the mean of that distribution. And we can describe the uncertainty around that mean by using either the standard deviation of the distribution or an interval. And then we can make probabilistic inferences about the classification for maturity of the animal often we use length as a way to classify animals as being mature or immature. And so we can create rules like if an animal's distribution is greater than 50% larger than the length cutoff, we can assign it as being mature or immature. So it has opportunities to kind of have more robust classification methods. And so I applied that to the 52 humpback whales. This is an example of a single individual. So on the right, we have the same individual. On the top panel is is model 1, which is just using altitude from the barometer to estimate length. And the bottom is using a barometer and a laser. And the black bars represent the uncertainty, which are the 95% highest probability density intervals, or HPD intervals. That represents the region with a 95% probability of encompassing the true length. The red line represents that maturity length cutoff I was talking about. And so the thing to notice that stands out is that the top panel using just the barometer has a much wider distribution, a much larger uncertainty compared to the laser on the bottom. And it also has a larger mean, that little point in the middle, predicted length, where this animal would be classified as mature on the top, but it would be classified as immature on the bottom. And so looking at the total 52 humpback whales I compared, again, we have model 1 on the top with just using a barometer, model 2 on the bottom using a barometer and a laser. We have predicted length on the y-axis. Each point represents the mean of the distribution for each animal, with the bars, those 95% highest posterior density intervals as the uncertainty. We have the observed altitude on the x, and the red line represents the maturity cutoff, and the color represents the probability of being mature. So lighter colors represents more mature, higher probability of being mature for those animals. And so just looking at top panel versus bottom panel, the first thing to notice is the lengths are a lot larger and they have much wider uncertainty compared to the bottom panel with the laser. And there's even misclassifications of animals as being mature and not immature. And so the laser just produces much tighter, lower uncertainty. It's much cleaner. And so the take-home message is, if you can, you should always fly with a laser altimeter. You'll get much better results. And so I then wanted to scale this up and try to, how can we incorporate this for measuring body condition of whales? And so body condition is the energy accumulated in the body as a result of feeding. And man, I feel like I should insert like a holiday joke here a week after Thanksgiving with that, but I'll let you guys come up with a clever joke. Here we define body condition as the morphological measure of an individual's relative energy reserves compared to its structural size. And it reflects an individual's foraging success and provides information on habitat quality and reproductive output. And so when we're measuring body condition in baleen whales, we usually use 1D, 2D, or 3D measurements. 1D measurements are single width measurement. 2D, we divide the length into perpendicular widths down the body to calculate a projected dorsal surface area or a body area index. 3D measurements take the same width segments, but then calculate a body volume measurement, and which can be converted into a relative measure for body condition index. Body volume can also be converted to estimate mass, which is really useful for energetic studies. But there's been no comparison of these measurements or how uncertainty scales across these multi-dimensions. And so we actually just published a paper last week doing this. And so I'm excited that that just came out and I can share those results with you guys. For this analysis, we use morphometrics again, which I just mentioned, to measure the body condition of the animal. We also then, Clara Bird and I, who's here in the audience, created collatrix, which takes morphometrics outputs and collates them into a single data file, but then also has a function to be able to automatically calculate each of those body condition measurements I just mentioned. So pretty awesome tool. Thank you, Clara. And so for our analysis, we measured 127 whales, 32 blue whales, 55 humpback whales, 40 Antarctic minke whales. We incorporated uncertainty associated with the different body condition measurements. We evaluated the scaling of uncertainty, analyzed the precision with each measurement, and evaluated the correlation between these standardized metrics. So when we have a morphometrics output, this is what it looks like. You can see the total length and then the width segments down the body. We first identify this head to tail range, which basically excludes regions of the body that's not known to store energy or lipids. So that excludes the head, the fin, and the tail. We then further develop the uncertainty model that I just mentioned to create posterior predictive distributions for each width. We then use collatrix to calculate each of those body condition measurements, and it does it for each sample in each width measurement, which results in 50,000 iterations, which then generates a posterior predictive distribution for each of the body condition measurements. And now we can actually compare the uncertainty between all these different measurements. And so this is the results. And uncertainty does not scale linearly across 1D, 2D, and 3D measurements. And that's what that looks like on the right there. We've got on the x-axis your 1D uncertainty. Uncertainty here is measured as the standard deviation of those posterior distributions. On the right, or on the left panel, we have 2D uncertainty on the y-axis, so surface area. On the right figure, we've got 3D uncertainty, so body volume. And the different colors represent the different species. And so uncertainty associated with 2D and 3D measurements increase at a greater proportion as uncertainty associated with 1D measurements. And so what this means is that there's an added cost as you move up these multi-dimensions and uncertainty. And this isn't to say, not to use 3D metrics by any means, because like I said, 3D metrics using body volume can be really useful for estimating mass, but it just highlights the importance of incorporating uncertainty, because then you can, to mitigate that as best you can, like choosing a certain camera or making sure you fly with a laser altimeter or flying at altitudes that will yield lower uncertainty. And so to compare the precision, precision is the closeness of agreement between independent measurements of a quantity without reference to a true value. We don't know the true value of the true body condition of the whale in this case, but precision can tell us if we're able to actually detect a small change in body condition. And so we use the coefficient of variation, or CV, and so lower CV equals higher precision here. And so it's pretty obvious that body area index, or BAI, has the highest precision. And so this means it should, in theory, be able to detect the smallest amount of change in body condition. And BAI was developed by Lee's lab in 2018 and is standardized by individual's length. And so to compare to other standardized measurements of body condition, all are highly correlated. So this is a really important finding because it shows that relative measures of body condition, no matter what dimension you use, 1D or 2D or 3D, should all produce a similar conclusion. And so researchers can be confident that they'll draw a similar conclusion no matter what standardized metric they use. But with that being said, there's still some major advantages for using BAI that we found. It's standardized per individual. It captures variation along the body. It has high precision, so able to detect smaller changes in body condition. And it's easier to compare across individuals and populations over time. BAI is similar concept, at least, as a body mass index in humans, or BMI, where there's this, we can get a number and it's kind of like a health score, and we can compare and know how healthy we are relative to the population. And so what's interesting here is you look at the different colors of BAI on the plot on the left there. Different species seem to have a specific range in BAI. So I think the next step is to kind of identify what is a healthy range for each species. And so that's something that I at least try to take one step closer to during my PhD as well. So I'll talk about this here, looking at the seasonal gain in body condition of foraging humpback whales along the Western Antarctic Peninsula. And so this is stock G humpback whales. They forage along the Western Antarctic Peninsula. They undergo one of the longest mammalian migrations, migrating all the way up to Colombia, even past the equator to Costa Rica, which is their breeding grounds. They're capital breeders, so they use stored energy to finance the costs of migration and reproduction. And they must acquire enough energy reserves once they're on their foraging grounds in Antarctica. And they do this by exploiting large abundance of krill and do that by adopting optimal foraging strategies. And stock G is a recovering population. It's estimated that between 15,000 and 20,000 individuals were taken between 1905 and 1968. Globally, though, humpback whale populations seem to be successfully recovering. This certainly seems to be the case for stock G. There's a high mean pregnancy rate reported between 2010 and 2014, with more than 50% of lactating females also pregnant, which means evidence of annual pregnancy, which is pretty crazy for an animal like this. And we think that this is attributed to a high abundance of krill available and lack of competition from other baleen whales that have not similarly recovered like they have. And so objectives with this study were to examine how body condition changes over the course of the foraging season and to describe how body condition changes for each reproductive class. And so for this, we used 3 different drones equipped with different cameras and focal length lenses. And we collected images during the austral foraging season, which was from November to June, 2017 to 2019. And it's difficult to get down, or it's challenging to get down to Antarctica. It's expensive, it's remote. And so we were able to work with NSF and work from NSF research vessels as well as Palmer Station. But we were also able to work with One Ocean Tour, which is a tour company that were awesome. They're unfortunately no longer in business, which is a shame because they were really great and really supported our science. And in case you're wondering if there's any differences between working from an NSF ship versus a tour ship, the answer is yes. In particular, when you go to the cafeteria, you sometimes are presented some mysterious food on the left there. And then strangely, with the tour ship, you're eating too elegantly for an Antarctic expedition sometimes, but hey, it's all good in the name of science. But both provided incredible opportunity to access the peninsula, a lot of different locations, different times of year. Got to see some incredible places, so really grateful to have those opportunities. But really importantly, they really filled the gaps in to be able to actually collect the data to answer a question like this. And so if we got the austral foraging season on the top from, we'll say November 1st is early season all the way to late June, you can see that One Ocean tour expeditions really filled in the gaps to be able to actually collect data across this entire season. So I'm really grateful for their support. And so this should look familiar. We'll measure each whale in morphometrics and identify its head to tail range, generate posterior distributions for each width, calculate BAI, and then generate posterior distributions for total length and BAI as well. We also collected biopsies samples to get sex and pregnancy. And we assigned reproductive classes. Lactating females and calves were determined by their relative size and close proximity to each other. Individuals were assigned as mature if greater than 50% of their posterior predictive distribution was greater than the minimum cutoff length for the maturity for that animal. So like I mentioned earlier, we use whaling data from Childerborough to determine what that length cutoff should be. And if we had the sex information from the biopsy samples, we could incorporate the sex-specific cutoff length at maturity. So for statistical analysis to be able to incorporate uncertainty, we calculated the variance of each posterior predictive distribution for BAI for each individual. And then we use weighted linear models to investigate how BAI uncertainty is influenced by year, time in the season, and reproductive class. We determined the most parsimonious model using ANOVA. And we used the best fitting model to describe how each reproductive class increases BAI over early, mid, and late season. And so trekking through the ice to the results. Here we have each box represents each reproductive class. So it's basically just the sample size for each reproductive class by month. Month is on the x-axis starting in November all the way to June. We had a total of 236 individuals, no repeats. So this represents a cross-section sample of the population. No samples in April and May. And we had about 509 images, which equated to almost 11,000 measurements in morphometrics. And so a huge thanks to Alison Dawn for helping me out with that. Speaking of Alison Dawn, I just want to say happy birthday to Alison Dawn, as well as Alejandro, who, what are the chances of having 2 people in the GEMM lab with the same birthday? That's pretty crazy. So if you see Alison, say happy birthday. I can see that on the right. And then, so some reproductive classes had low sample size across months, so we excluded them from the analysis. This was mature males. We only had 4 pregnant females, unfortunately. We had 1 lactating and pregnant female. And so these are the results. BAI had a curve linear increase rather than a linear increase. And so each plot here is each reproductive class. On the x-axis, we have days in the season. So starting from November 1st as day 0. We have BAI on the y-axis. Each point represents the mean of the distribution for BAI with the bars around it as those HBD intervals for uncertainty. And the colors represent time in the season, so the months in the season. So darker colors represent later in the season. And so BAI is best described by this orthogonal quadratic polynomial, which basically describes this curve linear increase. Each class showed this similar response, but there is differences amongst them. And there was no difference in BAI between years, between seasons, which was interesting. And so to better describe the differences between each reproductive class, we took the average whale in each class from the model and looked at what the expected BAI would be for that animal in that class on a given day in early, mid, and late season. And so early, mid, and late season here are on the x-axis as December 12th, February 8th, and March 26th. We have BAI on the y. The point represents that average expected BAI for that class and the 95% confidence intervals around that. And so each class has a much greater increase in BAI earlier in the season compared to later in the season. And so why is there this curve linear increase where they seem to not put on as much weight or gain as much body condition later in the season? And it might be where fat is deposited in the body. Sliper in 1962 noted that in the beginning of the Antarctic foraging season, fat is first stored as blubber, but then after January, it is deposited mostly in the skeleton, muscles, and the viscera. So this actually lines up with our timeline pretty well. And depositing fat into blubber first may optimize energy storage, and it might prevent overheating and excessive buoyancy. And a study earlier this year by Aoki looked at body condition measured from drones and found that increases in body condition was directly linked to increases in buoyancy. So it's possible that through photogrammetry, we're actually able to capture this transition of fat being deposited into the blubber, into the skeleton, muscles, and viscera. This similar trend was also noted by Rob Williams on North Atlantic fin whales, where blubber thickness had this asymptotic relationship where it increased and then kind of remained stable for the rest of the foraging season. While not explicitly described in gray whales, there's clearly a similar trend with BAI increasing and then plateauing later into the season as well. And this has also been described in hibernating mammals where body mass will increase and then plateau just before hibernation. And so if we look at a schematic of seasonal fattening for a pre-fasting mammal, we've got season on the x-axis, and then we have body mass in blue that will increase and then kind of level off just before fasting, whether it's migration or hibernation. And while food intake and body mass are positively correlated, food intake will actually peak during this hyperphagic phase and then decrease before body mass reaches its max level. And a study by Ross Nichols, who worked with Ari Friedlander at Santa Cruz, studying the same humpback whales that I'm studying, or that I was studying for this analysis, he was tagging them and found that foraging rates significantly decreased from summer to autumn. And so it's possible, or it seems like that humpback whales actually may be following this same trend where food intake increases and then declines, and then body mass follows this curve linear increase later in the season. And so comparing reproductive classes, lactating females had the poorest body condition, reflecting the high energetic cost of lactation. They displayed the greatest increase of BAI from early to mid-season, but they still had a significantly lower BAI compared to mature animals by late season. And then mature animals had similar levels of BAI by the end of the season, likely reflecting the high energy stores that they'll be using for reproduction. Immature whales had an interesting trend where they increased that, they had the similar trend where they increased BAI, but then it actually declined later in the season. And so this could be a result of sampling skinnier animals. Avila et al. reported that arrival times to their breeding grounds in Colombia are now 1 month earlier than they were 30 years ago. And they speculate that most of these early arrivals are immature whales. And so it's possible that immature whales reach a BAI level reach a certain level of BAI by mid-season, and then take off to the breeding grounds. And there's this catch-up period later in the season for whales that haven't quite reached that yet. And that's what we were observing. For calves, this is presumed to be their first season along the peninsula. And they also showed the similar trend of increase in BAI. But they did not show any increase in total length, which is the opposite of what was observed on the breeding grounds by Frederick Christensen. And so we're thinking that this might be a strategy to reduce heat loss by having this switch. And I threw this in here to kind of tie this in together a little more with the results I just showed you. This is the number of voyages to Antarctica per year. So we've got time on the x-axis all the way from 1992. The lighter color is number of tour ships that go to Antarctica. The darker color is NSF ships. And as you can see, NSF ships for the past 30 years have kind of remained pretty constant, where there's been this huge increase in tour vessels. And so, and I don't know what it stands now because of COVID, but last time I was down there, there's plans to bring in 30 more tour ships down to Antarctica to be part of this. So this is expected to grow even more. And so I think, being able to have this understanding of when humpback whales put on the most weight, when they gain their most BAI, could be useful to restrict whale watching vessels, earlier in the season compared to later in the season, when that's a crucial time for humpback whales to be putting on mass. Another thing to consider with this, looking at this, is this, with NSF not increasing opportunities to go down to Antarctica, which is ironic because Antarctica is one of the fastest growing or rapidly changing environments. Maybe there's more opportunities for us to work with tour operators to actually have access to these places that are changing, to actually get data. And so we kind of take some thinking outside of the box. So bringing it back to where we are now, like I said, started working with the GEMM lab this summer, and it's been awesome working with this group. We've been studying the Pacific Coast feeding group gray whales that forage off the coast here between May and October. And the project that comes to mind is the Granite Project, which is gray whale response to ambient noise informed by technology and ecology. And we're taking this non-invasive, multifaceted approach, collecting photo ID to link to sightings history, collecting fecal samples for hormone analysis, looking at pregnancy and stress hormones, using drone flights, looking at behavior and body condition changes over the course of the season, and collecting prey samples, looking at abundance and distribution changes. And then also we have hydrophones collecting, being able to quantify the soundscape as well. And so really excited to be able to, we're all in the thick of analyzing all this data we've collected this summer, which I'm really excited to then integrate into this framework. And this is just a quick view of, this is just the drone body condition measurements. I'm in the middle of processing measurements right now, but we've got animal ID on the y-axis for 2020 field season and 2021 field season, and time in the season on the x-axis. And the colors represent time in the season, so lighter colors are later in the season. And the thing that stands out is we've got several animals with repeated measurements over the course of the season. So we'll be able to look at not only just the body condition change of a specific individual over the course of the season, but we'll also be able to look at hormone and behavioral differences, and actually start to ask questions, what is the expected normal increase for BA over the course of the season for a typical PCFG gray whale? How is this influenced by hormones and pregnancy, environmental variation? These are just for 2 years, but we've got data from 2016. And how does this change, or how does behavior change with this as well? And we can then start to ask, what is a healthy BAI range? What BAI should a whale be by a certain time of year? And can we identify pregnancy via photogrammetry from drones? And can we predict potential strandings or future UMEs? If there's a higher proportion of animals below a certain BAI level during a certain time of year, would we then expect to see more strandings the following year or a lower reproductive rate? And so to conclude, developed the Bayesian statistical model for predicting measurements and associated uncertainty of unknown sized whales based on drone platform and altitude. Provided the first comparison of different body condition measurements and how uncertainty scales across these multi-dimensions. Use uncertainty model to demonstrate how BAI increases curvilinear for healthy humpback whales, and gave a little quick view of the next steps for integrating this framework through the multifaceted non-invasive research approach that we're taking in the GEMM lab on the PCFG gray whales. A lot of people to thank. So one of my favorite things about science is it's a team sport. Cinnamon hosted the research summit on collaboration, and there's so many things that go into and so many parts that go into a project. So a lot of people to thank here. And with that, yeah, I'll take any questions. Thank you for your time.
All right, so if you're in the room and you have a question, just raise your hand. If you are online, please go ahead and type it into the chat box or raise your digital hand and we'll get to you there. So I see we have a question in the chat box. I'm going to hand it to Michael.
So this is a question from a guest. What about non-orthogonality of the camera? Do you assume you're always positioned exactly above the whale and looking straight down?
Great question. Yeah, so how do you ensure that the camera is facing straight down as you take an image? So we put the camera on a gimbal, and so even if the drone is wobbling around a lot in the wind, the camera will always be straight. And we have a way to tell if to make sure that the camera is actually facing down when we take the photo as well. Because a lot of times we'll have it up as we search for the whale, but then we can control it to make sure that it's facing straight down once we're over the whale.
Great questions in the room. I thought I saw a hand. Hang on one second.
Thanks. Great presentation. Just truly amazing work and really well presented. First, a comment. Your arrows on the top, like were on your second slide, many of them seem like they could go both directions, right? Mm-hmm. So it's not necessarily a one directional sort of thing with morphology or morphology going to one direction, but I think it might the ecology of the animal also shapes the morphology as well. But what I wanted to ask about was altitude was a really important thing in there, but how much underwater are they and how do you deal with that level of error as well? Because water clarity can make a big difference. And, so if they're, a few meters, it's, it maybe it's not that much of the proportion of altitude, but it still seems like it might be something to consider.
Yeah, great point. And thank you for the comment about the flow chart. That's a great suggestion that I'll need to revamp. But yeah, great point about the depth of the animal as well, because if the animal is actually deeper than you think it is, we're assuming that it's at the surface, but if it is deeper, that's actually adding altitude, which will change your scale technically. And there's also parts with refraction as depth increases as well. And so that's a really challenging thing to get at. So we try to, one thing is to first filter photos where it looks like the animal is at the surface or just about to break the surface, and you can tell, from before or after the photos are taken to kind of help determine. And so that's one aspect that we can do that's quick and easy. Another aspect is, and I've been thinking about this a lot, and I might kind of bring it back into light to kind of to do a study with it, is from some of the work I've done with Jeremy Goldbogen's lab at Stanford, where we've tagged whales and flew drones over them, we can actually use the tag to determine how deep the animal is. And so it'd be great to do a control study where, okay, I confirm that the animal's at the surface straight, and then measure it when we know it's at a specific depth from the tag, and then compare how that influences uncertainty. And that would be easy to then incorporate into the model as well. The other thing is I have some data that I did during my PhD that I didn't end up doing anything with, where I made this control study where I got in a pool and I had these known size objects at different depths of the pool. and I had a camera above it at a known height, and I created waves and I measured the height of the wave to be able to determine the refraction. And it's really cool data. I just didn't end up doing with it, but you're kind of making me want to get back into that to see how refraction impacts objects at different depths. So great point.
Great. Another question online.
This one is from Rich Morgan. What would it take for a DSLR aerial photograph to be useful? Possibly connected to an altimeter so that the photo could be tagged when it is taken.
Possibly connected to what was the word?
To an altimeter. So
Altimeter. Yeah.
altimeter.
Is this like from, I'm assuming just like opportunistic photos from above. Yes, if there's any photos from above of a whale, I'm assuming this is the question. What would be useful to be actually help with the scaling of that? Any sense of altitude would be really helpful to get an idea of to set the scale of the photo. And if it's off, if it's not quite nadir or straight down the camera view, we can try to do some trigonometry to be able to adjust that. The good thing about BAI too, which I didn't mention, is, and we're doing this in a study where we have opportunistic data of Arctic gray whales from a flight that didn't have altimeter information, so no altitude. But you can still get a BAI measurement because it's scale invariant. And so you can actually just apply it just by looking at pixels without ignoring altitude, and you can get a similar metric for BA to kind of get a rough estimate of what the animal's body condition is, which is a really valuable aspect of using that too.
Great. Any questions? There we go.
Thanks, Casey. So I'm interested in the curvilinear response of those whales and Antarctica. So this is kind of maybe like a dissertation defense question, but if you only could go down to Antarctica during 1 month of the year, okay, and you wanted to assess like the health of the population, you know, based on these changes in their foraging rates and sort of the delayed increase in body mass, and that might be due to different things. If you just wanted to know how healthy a population would be, can you, based on what you know now, pick a month that would be best to go?
Are you asking for me to suggest a month? Or?
Well, I'm just asking you to incorporate all the information that you put up there about what we know about these capital breeders and how they put on mass based on their feeding. If you wanted to measure healthy whales, the healthiest time of the whale, when would you go?
Yeah, if you could get, I mean, if you could get at least 2 time points in the season, I think that would be really valuable. But if you only had one, let's say you only had one, you get down there, when would you go? I would probably go, not necessarily in the early season, I'd go just after it. So just later, right before March, right before that last time point I saw, because that gives a grace period to see if they can reach that asymptotic range in BAI for each reproduction. Yeah, exactly.
All right, we got a question online, and then I got another question in the room. So start here.
This is actually an in the room question from me.
Oh.
So I would like you to remind us what BAI is and comment on the fascinating thing you shared, that the different species seem to cluster differently for BAI, and what ideas you have for why that might be.
Yeah, that's a great question that we definitely could have spent a lot of time talking about too in the presentation. So BAI is body area index, similar concept as BMI in humans, body mass index, but we use area as a surrogate for mass. And so, you know, similar in humans, if you are between, I think it's like 18.5 to 25 BMI, you're considered healthy. And so this idea of, you know, could we measure a whale maybe during a certain time point, and is there a healthy range of BAI for each species? And it seems to be species specific, and they seem to be clustered. And so I think this has to do with body shape. It's interesting, and I have a plot that I didn't include here, but those different species I included, minkies, blues, and humpbacks are all very different lengths, very different body shapes. And so the great thing about BAI is it accounts for those different body shapes to be able to kind of get at what a healthy range would be. That answers your question.
All right, question in the room.
Hi, thank you. Really great talk, really well presented, interesting science, very nice. So my question is, it looks like we are getting pretty high precision in measuring the size and the body conditions of these animals, but they're still pretty large and slow. So my question would be, how far are we to get that level of precision in smaller animals, faster animals, for example, smaller odontocetes? And my second question is, if you saw any difference in BAI or BMI and the researchers from one type of cruise to the other, from the cruise ship eating the fancy food versus the other eating the creepy food, if there's any difference along the season.
Big difference. Yeah, so the question is, can we essentially apply this to smaller, fast-moving cetaceans, maybe dolphins or porpoises? The answer is yes. And Julian Dale, who I worked with at Duke a lot, who was helped pilot for some of this data, he's doing that, or he has a study coming out actually doing that with Scotland dolphins. And so you just might have to change your flying style. You might have to change the shutter speed of the camera. Maybe you record video instead of taking photos to be able to capture it. But yes, definitely. And so it just, yeah, takes a different approach for flying, but absolutely. Yeah, and then so, yeah, the cruise ship is pretty interesting. It's funny going on these epic Antarctic expeditions, you know, out in the cold, and then you come back and there's like hot cocoa served, and you know you got your Patagonia jacket, and yeah, pretty interesting differences between those.
Great. Question online, Michael? I know we've got the messy screen because of how we have it set up, but.
Oh, it's gone. This is Ryan Stevenson. Have you been able to use drones to measure the krill concentration that baleen whales feed on?
Ooh, I love that question. In not this photo, but another photo I had of bubble nets, bubble netting by the humpbacks, there is a cloud of krill that you can actually see in it, and you could measure. It just obviously gets difficult with, you know, the depth and thinking about that. But it would be cool to see, yeah, if we can measure if the area of these plumes of krill essentially. It's not very reliable over time, but there certainly are cases now and then where we do see them at the surface. Most of the time we don't see them at the surface, but there have been a few times. So that could be a good opportunistic sample that you can get from that.
Great. Any other questions in the room? Any other questions online? All right. Thank you everyone for being here, and thank you, Casey, for presenting. We really appreciate your time. For those of you in the room, you can come on up and talk to Casey if you have additional questions. For those that are online, I hope we see you again next week. Thank you, everybody.
Thank you.
Okay, for those online, we're going to end the presentation now. Thank you so much.
Not perfect. No, it's